Greg for joining us. Just like to introduce our speaker. Dr. Gregory Kiar has a master's in biomedical engineering from Johns Hopkins University and recently completed his PhD in biological and biomedical engineering at McGill. He is presently a research scientist at the Child Mind Institute, where his research continues to focus on evaluating and improving the trustworthiness of tools and techniques used to study the brain, with an emphasis on biomarker development. Greg tackles these issues from a variety of angles, including by developing computational infrastructures that support reproducible neuroimaging experiments, evaluating the role that unavoidable computing errors play in results, and leveraging such errors towards building robust models of the brain. So Greg, would you like to have me stop you and ask questions during the talk, or would you like to leave the questions to the end?
Absolutely throughout. There'll be a few times when I ask for input. So people, if I'm asking one thing or another, you can use the reactions buttons. So familiarize yourself with where they are now.
Right. So definitely do that, everybody, and feel free to write in the chat or raise your hand during the talk. Go ahead.
Cool. Thanks. So yeah, thanks for the introduction. So as Katie said, there we go. Now my mouse is back. Yeah, so I'm a research scientist recently at the Child Mind Institute. Today I'll be talking to you about how we can kind of understand the role that numerical errors and uncertainty have in our scientific results, in particular with my use case in this presentation being network neuroscience. So the concepts and results can really apply to any computational domain. So I'll start by just introducing the concept of reproducibility. So reproducibility, as I'm sure many of you are aware, is really just being able to generate the same finding multiple times, whether this is from different data sets, slightly different tools, or exactly replicating analysis. There's different terms and subterms that have all kind of have fuzzy definitions around this, but the core concept remains, can a finding be regenerated? Now, unfortunately, this has been shown recently to be anything but a guarantee. In 2015, a group called the Open Science Collaboration attempted to replicate 100 studies in psychology and found that nearly 2/3 of them failed to replicate with a classically significant p-value of 0.05 being the threshold. This is unfortunately a pretty significant portion that failed to replicate. And these issues aren't just in psychology. There's an infamous paper at this point called Cluster Failure that came out a year later in 2016, which basically showed that various software bugs in some popular image analysis libraries for fMRI led to incredibly inflated false positive rates. So sometimes as high as 70% when the significance threshold was set to be 0.05 as well. So unfortunately, reproducibility may be an issue anyways. So we're going to start by talking about some places that may contribute to this. So for one, we have tool selection. In the last couple of years, there's been quite a few studies which have explored basically the impact of choosing a different tool or a different technique to study similar things. So on the left here, we have a paper by Alex Bowring, which basically took 3 published analyses using data sets hosted on OpenNeuro, and found that using different libraries, you end up with considerably different statistical maps. Now, to a whole other scale, this was really replicated both in fMRI again and in diffusion, where in the diffusion case, I think it was 48 teams, and in the fMRI case, it was 70 teams. Were essentially, in both cases, given the same data set and told to answer the same question. And it was found that there's a huge amount of variability in these results. So basically, tool selection and the decision-making scientists may, in some cases, contribute to some of the variability or lack of replicability that we may see. So next fact. So sometimes it could be something a little simpler. It could be simply what operating system you're using for your computer. In this case, what we're seeing is the Human Connectome project, pre-free surfer pipeline, I believe, processing a subject from the Human Connectome project data set. So, very high fidelity data, very high quality pipeline. And the flickering you see is between, does the flickering come through, by the way? Yeah, perfect. The flickering you see is between just running one version of CentOS, the operating system, and another. Not even, a really old one and a really new one. Both of these are commonly used. And really, it's one core math library that's causing most of these differences. But if you care about things like, your gray matter, white matter boundary, especially in regions like this can be a pretty alarming difference. So let's see. Now we know that our systems may play a role. Well, sometimes also, say we're trying to replicate out of data and there's slightly different noise properties or what have you. Well, small, ideally insignificant amounts of noise can also play a role which prevent us from replicating. So in this case, Lindsay Lewis in Alan Evans's lab, she perturbed one voxel in structural MRI in the T1 in the corpus callosum. She made it 1% brighter. So if the original intensity was, say, 100, she made it 101. Then she reran FreeSurfer and Civet to generate cortical surfaces. And everything you see that's not green is how these surfaces changed from these two different runs. So again, these are gray matter surfaces. And she perturbed one voxel within white matter by 1%. And we see pretty significant differences, just because of this small amount of noise. So sometimes we have noise properties, we have system properties, we have which tool we used. Well, how can we start to, actually understand where this is coming from? And so one thing where you can start to see a little more concretely where this may be emerging from is when you think about algorithm selection. So if you have a bunch of competing algorithms that can all do the same thing, some of them may be more stable than others. So what we see here is that for different tensor reconstruction algorithms, you can see that the theoretical condition or the theoretical stability of these tools is very, very tightly related to the variability we observe in practice. So again, in this case, you expect the tool to be less stable, which means more sensitive to noise, and it is. And again, the ones down here are indeed less sensitive to noise. So sometimes you can pick a different algorithm and some will just be better and some will just be worse, and you can anticipate this. Not always, but in theory, you can. So this is where it gets a little discouraging, and I promise I'll be encouraging as the talk goes on. But unfortunately, computers are imperfect machines. So in this case, I opened Python 3. Any of you could do the same thing on your computer. I wrote a little for loop that basically what we're doing is we're adding the number 0.001 1,000 times. This should give you the number 1. It's a pretty simple operation. You can't make a pipeline much simpler than that. What we see in reality is there's a bit of error because computers are imperfect machines and precise machines, and there's some error in how the number 0.001 is represented. So then this error can cascade and accumulate over operations, and you can end up essentially losing information. Now, this is an unavoidable problem in the sense that even if you were to have bigger data types and store data in larger containers, you would at some point always be hitting these thresholds. So this is going to be an exercise where it's going to be a bit interactive. So one example of how these errors can cause a problem is really through any arithmetic operation that doesn't have the same properties that we would expect when we're considering real math arithmetic versus floating point arithmetic, where a floating point is just a decimal number. So for instance, addition is a non-associative operation from the perspective of a computer. So we have two ways that we are ordering this expression, which I think we would agree if we're doing this just by hand would give us the same answer. But the trick is that we have 4 digits of precision. So never, and when you're performing this, can you keep track of more than 4 numbers at a time. So here's where we're going to do a reaction game. Everyone raise your hand if you, in the chat, if you think the top answer is the right answer. No one? I mean, maybe people haven't found the button. Okay, how about the bottom one? Who thinks the bottom is the right answer? No reactions. Oh, I see a hand. There we go. Okay, who thinks neither is the right answer and this is a trick question? Katie as well, that works. So we're gonna go through it and talk about 2 types. Can't read this very well. Oh, sorry, Gabe. So basically in the first case, we have 2000 subtracting 1998. What this does is it gives us the number 2. Now, once we have the number 2, we then do our addition, we add 1.333, and here we go. Now, the problem here is that essentially we took two numbers that have 4 digits of significance each, because, in this case, we know all of these digits in 2000, we have certainty in them, same in 1998. And we end up having a number where we only have 1 digit of significance. So again, here, we don't know anything about these zeros. We don't know if they're actually zeros because we don't have any information from the 2 original numbers. So we can't actually say with certainty this is 2.000 versus 2.4, maybe it's 1.9. We can't really say because we don't know what they are. So then when we add this 1.33 at the end, we really don't have confidence in anything after the decimal. So this is what's called catastrophic cancellation. We take things with a lot of signal and we end up, just because of the way we did our math, we end up reducing the amount of signal in them. So there's another source of error we can see. Excuse me. So in this case, we're doing our negative 1998 plus 1.333 first, which, because we can only keep 4 our digits of significance, leaves us with negative 1997. By the way, Millar, do you have a question, or is just the reaction still up?
It's a vestigial hand. It's from before. Yeah.
Perfect. No problem. So in this case, what we've done is we have the number 1997, and we've lost all of these 3s. So we've done some round off error here, and now we don't have the same precision we had before. So then we're doing this new sum of 2000, subtract 1997, and we end up with 3, which also, by the way, is another example of catastrophic cancellation. So we end up having not a good sense of the actual significance. So in both cases, our answers are just estimates. They're both biased in some way that just happens to be different from one another. So now, considering bias, one thing to think about is the bias variance trade-off. So this is a concept that's been around in machine learning for quite a while, and it basically says that that as you have either a lot of bias or a lot of variance, your models will generally be wrong. However, there's a tipping point between the two. As you're getting rid of bias and adding variance, and vice versa, that you will end up having less error in your total models. So in other words, you'll have a bit more accuracy in what you're doing. So what we've seen here is that we have bias in our results. So what I'm going to talk about for most of the talk, and I'll get to in a little bit, is how we can turn this into variance instead. So one other example just of how variance can be a useful thing. For anyone who's familiar with machine learning, you may be very familiar with a figure like this. This is a random forest. It is a type of classifier, which really kind of embodies this concept of trying to capture variance. Basically, you randomly generate slightly different classifiers by truncating your data and truncating your samples, form a bunch of independent simple classifiers, and then ultimately, by considering them all at once, by considering all the variability you've sampled, you can create higher fidelity predictions than any one of these classifiers could on their own. So this is a really strong and popularly used classifier that really embodies this, again, this concept of trying to capture variability rather than pretending it isn't there. So I've just started by giving you guys a bit of a background on what I'm talking about when it comes to reproducibility, variability, and kind of floating point uncertainty. So I'll give you a second to read that, and then I'll talk about what's next. So the next thing I'm going to introduce is a concept called Monte Carlo arithmetic, and a library that I've made using it called Fuzzy. We're going to talk about how this can be applied to perturbing their imaging pipelines and capturing a bit more variance in their execution. And then we'll see how I can use this variability to actually augment our data set and improve the quality of some models. And then finally, I'll talk about how this can be kind of applied more generally to other areas of neuroimaging analysis, or any analysis, really. So Monte Carlo arithmetic is a technique which basically takes your floating point arithmetic and adds a little bit of error to it. So I'm not going to bore you with digging into the equations, but I actually made a pretty figure instead, which makes it hopefully a bit more digestible. The concept here is basically for any floating point operation. So Gabe said he's having trouble reading with the slides. I'm guessing on a small screen this isn't great, so I'll walk you through with the pointer. Okay, thanks, Gabe. So basically every floating point operation, so again, mathematical operation using a decimal object is in green. So what this tool called Monte Carlo does, which basically implements Monte Carlo arithmetic or MCA, is it basically says anytime we have a decimal operation, we are going to add this little workflow, which allows us to add some noise to our operations. So we'll pretty much add noise wherever we want it, perform our operation, maybe add a bit more noise depending on the configuration, and then bring their data right back into the normal space. Now, to talk about this at a higher level and conceptually what this means is it allows us to basically make the error that's associated with our computers be probabilistic. So instead of me running that Python loop that I did in the first, in one of the early slides rather, and always getting the same wrong answer, the 1 point, a bunch of zeros and a 7, it'll give slightly different wrong answers each time. Now the concept is that if you do this a number of times and you average all your results, your average will be closer to the true answer than any of the individual biased answers. So essentially what you're doing is you're adding variability in how this bias is affecting each operation or each evaluation of your pipeline. So then you can essentially then capture it and have estimates that are aggregated with less bias. Does anyone have any questions about this before I move on? Okay. So again, basically the place I'm doing this here, when we showed that little example of how we had uncertainty in our trailing bits. Oh, is someone unmuted? Not sure if that's intentional. Okay, he's muted again. So essentially what I was referring to earlier with our addition example was basically that we'll have a bit of loss at the end of our results. So what we're essentially doing here is we're perturbing the end of our precision. So basically I'm not perturbing anything that we know and trust. I'm basically making it so that last bit that we have in our data, the one that we don't really have confidence in because we don't know anything beyond it, I'm perturbing that one. And basically what we're saying is that means the results of my operations will be no more or less valid than what you had before without me doing these perturbations. Basically everything will, instead of being wrong in the same direction, again, it'll just be wrong in slightly different directions. And I know I've said this a few times, but I'm really trying to make that point clear because kind of the rest of the stuff I'm going to talk about relies on it. So the nice thing with a tool like this is you can instrument lots of different libraries with this type of logic, recompile them, and then have them ready to use. So we've set up things like libmath, which is used by FSL, FreeSurfer, AFNI, Octave, SPM when it's in Octave, BLAST and LAPACK, which is used in Python's NumPy and SPM, and the entire Python scientific stack, which is used by tons of tools. So the application that I'm going to be talking about is structural connectome estimation. So here we have diffusion MRI images and a parcellation. In this case, I used the DKT parcellation, the 83 nodes. And then we're going to run it through DiPy and generate a connectivity matrix. For the sake of this experiment, I didn't perturb pre-processing. So the data have been denoised and aligned and all of this stuff prior to any of the perturbations. The reason for that is one I'll get into a little bit later, but basically there can be a significant computational overhead with this process. So I didn't add that just because it already took about 20 years of computing, which thankfully I did in parallel. Otherwise I wouldn't be graduated. So yeah, it's generally an expensive technique. So then the experimental design is really, we have 100 subjects. We did our denoising and everything, as I mentioned. We do our modeling using a deterministic and a probabilistic pipeline in DiPy. And we ran these, excuse me, 20 times each, where all we're perturbing each time is the way that error is handled in the machine. Again, in no case is the error going to be more or less valid or accurate. Essentially, all we're doing is making it wrong in slightly different ways. So we end up with a ton of connectomes. And the goal then is to look at all these connectomes and see if we can capture the variability, or if the way we capture it is meaningful. So let's start. So there's two ways that I instrumented the pipelines. So one that I'm going to call dense MCA. In this case, I perturbed most of the operations in the pipeline. I instrumented as many things as I could. In the case of sparse MCA, I perturbed much less. The reason for doing the sparse MCA is it's much less computationally expensive because any single decimal operation or floating point operation takes place using Monte Carlo arithmetic, it takes about 100 times more time to perform. Now, thankfully, our pipelines aren't just floating point operations. There's tons of data handling and moving around and stuff like that. So in practice, the dense pipelines take about 7 to 10 times more time. But if we could get similar results or value from doing sparse perturbations, that would be much better from an efficiency standpoint. So now we're going to go into the results. What we're looking at here in these little box plots is as you go, as the points and boxes go further to the right, we end up with essentially a larger difference in our given connectomes from their unperturbed counterparts. Now, we can compare the variability we're looking at here across a few different axes. So we can look just across within the same data. All we're doing is looking across my MCA perturbations. So in the dense case, we actually notice that, that there's not a whole lot of variability we're having. We can, in some cases, reach the level of within acquisition variability, where in this case, what I've done is I've subsampled the data so that we have essentially even diffusion directions and odd diffusion directions processed separately. Basically, the reason is so we can have again, somewhere in between like cross-section level differences and just MCA. So this is essentially within acquisition, but separate subsamples of data. So in some cases, we see that the Monte Carlo can approach this in the dense case. But in general, we kind of have what we'd expect. We have the most variability when we're looking across subjects, less, but still pretty close, unfortunately, when looking across sessions, and much less when we're looking kind of within the same acquisition. However, when we look in the sparse case, what we can find is quite a lot more variability. And I can give some intuition to this a little bit later. But basically what we're seeing in this case is that our variability that we add, so again, this is in the purple traces, this is only comparing, my brain, my same piece of data generated with deterministic error versus the exact same piece of data processed with probabilistic error a number of times. And what we can see is we often have huge differences in the range of cross-subject level effects. Now, this is potentially very significant because we have the exact same data, and not only does it maybe look more or as much like a different session from myself as it looks like my own brain, but it could look like an entirely different person, or it's on the same order of magnitude with these differences. So that's potentially very concerning. So the next thing is to try and see, in general, what part of these networks is giving us this variability. So again, we have dense on the left and sparse on the right. That'll be consistent for the whole presentation, but I think I still annotate them. So here we've computed the number of significant digits for each edge in the connectomes, again, across these different groupings. So significant digits essentially means the number of decimal digits which are completely unchanging by all these perturbations. In other words, there's no variability in an edge at all if it's at 16, and if it's at 1, basically it's always changing. So what we can see again in the dense case is, in general, across MCA samples, most of our edges are pretty stable. However, when we're even looking across subsamples, so again, this is the same data, the same acquisition, just slightly different subsampling of our data, that degrades very quickly to around 4. And in the sparse case, we never get higher than this. So in general, at least in the sparse case, what we found is that edge weights are quite unreliable. And so one of the next questions is to try and see if the variability we're looking at across these different perturbations, again, the pipelines are generating equally valid results every time. So we want to understand what is the structure of these differences. How can we represent the types of changes or understand the types of changes we're observing? So what we did here was we tried to explore exactly that. We took a single reference graph and looked and just randomly sampled other connectomes that were perturbed and annotated them based, or sorted them based on their difference from this reference. Now, in this case, what we see from the variability is that our networks are very systematically and structurally different and degrading across these perturbations. What we see in some cases is, a graph near perfectly the same. And in other cases, what we notice is there's pretty much a collapse of connectivity within the left hemisphere and eventually across the entire graph. So what this is saying is that our pipeline is perhaps teetering on a bit of an edge for this particular subject, in that, again, from equally valid estimates of connectivity, in some cases we'll have a typical well-connected graph. In other cases, we'll be missing an entire half of it. And in some others, we may have basically no connectivity at all. So this kind of led us to believe this could potentially be a proxy for quality control, and something that we can look at in the future as well. But in practice, the way we're looking at network structure in general in network neuroscience is through, well, network statistics. So what we did here is we tried to look at the stability of various network statistics from these graphs. So again, in the dense case, we're very close to this top line here, where in a perfect world, all these bars would be touching that dashed red line. So in the dense case, again, in general, our statistics are pretty stable, where our least stable one, is certainly in the case of the deterministic pipeline, is edge weight. And because these are multivariate statistics, we're actually computing the stability of their moments, well, the first 5 moments, as shown here, the sum, mean, variance, skew, and kurtosis of these features. But again, when we look over to our sparse case, what we can see is that there's basically no trustworthy information in these networks. In other words, our statistics will be changing dramatically from one perturbed execution to another. And again, in some cases, literally 0 or 0 point some significant bits, digits rather, where in other cases, it's a bit higher, but still very low. So what this is saying is that individual network statistics for, person A has a clustering coefficient of blank, plus a clustering coefficient distribution of blank. These in general are very unreliable. Now, an interesting thing we see, however, is that the distributions of these results is very stable. So while, again, the clustering coefficient, for example, oh, the cat feeder always interrupts my meetings. Sorry if you heard that. It'll interrupt us twice more. But interestingly, what we can see is the distributions of these statistics is stable across an entire data set. So while, again, my clustering coefficient may be unstable, and Katie's may be unstable, and Millar's may be unstable, in general, the data set that includes all of us will have similar properties. Now, this is super interesting because what it shows us is that not only are we introducing this variability that is telling us that, okay, these individual estimates may not be as reliable as we'd like them to be, but it's very clearly showing that they're all remaining in the realm of the same kind of data set space. We are still sampling data from the same, again, biologically plausible, if you want to call it, or at least familiar space. So when we get back to the concept of bias, one important thing to consider is test-retest type experiments, and in general, the difference between sessions. So in a perfect world, a test-retest experiment would be kind of structured as follows. You have some number of subjects, you have 2 or 3 years or however many measurements each, and then you'll process all your data, and at the end of the day, you will compare all of your data together in a pairwise fashion and find that the observations that are always closest together are those that are coming from the same person. So in a perfect world, that would be the case. If that always happened, your score by most of these measures would be 1. And in this case, for the measure I'm using, which is discriminability, that would be quite a bit lower. Now, if we flip this on its head a little bit, we can think about things like, instead of using, individual as our labels, we can think about using maybe session and subsamples as our labels. So pretty much what I'm trying to say now is, if I have one person who has been scanned twice, and I have many observations of the connectomes from each of these scans, can I detect which scan I'm coming from? Can I tell the difference between scan A and scan B? Similarly, can I tell the difference between subsample, even versus odd? In a perfect world, our answer would be no. We want to have data that's reliable, that's consistent across these acquisitions, across subsampling, where there's no, reason the data should be different. So in a perfect world, we'd be getting something close to chance. We'd have no discernible way to tell them apart. In practice, however, what we can find is that, at least in the experiment that we could do in the unperturbed samples, there's always a considerable difference from session. Session does make a big impact. You can very consistently tell them apart. However, in the case of our sparse MCA case, we're actually reducing this bias a little bit. By adding variability in how our data is processed, this bias shrinks. And the same is true when we compare, again, our sparse case to the dense in the across subsample setting. So basically, these numbers are getting lower. And again, what that means is we're reducing the bias of session and of specific subsampling of our data. Now, this is potentially encouraging, because again, I told you I would eventually become a positive person, while the rest of this talk has been maybe scary. And really what we're trying to say now is, okay, if we have this variability that's removing some measurable form of bias, can I then capture this variance and maybe do something good with it? So what we see here is we're trying to basically describe this variability. Where here there's a simple classifier that was trained to predict high versus low BMI. And basically in the orange dots in the center of each of these distributions, you have your reference performance. Now, the distributions you see are actually the distributions using randomly sampled networks from all of our perturbed settings. And what you can see is the performance really ranges. So in some cases, yes, it's worse. Maybe we have worse quality data. The networks don't contain, quite as much meaningful signal, at least by this measure. And in other cases, it's quite optimistic. It's actually better. So really, instead of having these orange dots, which are basically point estimates of the signal of our brains related to this feature, we have distributions of them. So really, the next goal is to say, can we augment our data set through these Monte Carlo perturbed data samples, rather, to actually improve the quality of classifiers? So basically, I took this data set, did several different pre-processing strategies, such as taking like log transforms and ranks and what have you of networks, several different dimensionality reduction techniques, including PCA and feature agglomeration, and several different classifiers, which you'll see the list of them on one of the next slides. And basically, the special sauce, the magic bit, was right here, where we're trying to resample a data set according to the perturbations to basically see if we can capture any more signal by considering distributions of results rather than single reference unperturbed results. Does anyone have any questions about this kind of layout before I move forward? Cool. So the next thing that's important to understand before we move forward is the various ways that we're going to try and aggregate our data set. So there's a few different schools of thought. The first is basically saying, oh, I'm sure that variance isn't meaningful. I'm sure it's all garbage. So let's just remove everything that is variable at all. We'll only keep the pieces that are entirely constant. So sure, we can make an approach for that. This is the truncate method, as you'll see referred to later. And again, in this case, we're just removing everything that changes. So if, again, in one network, let's say we have an edge that's intensity is 130 and that ranges from 130 to 136. In this case, well, we'd only keep the significance corresponding to the 1 and the 3 because we don't have certainty on that last digit. So basically, in this case, again, you're just keeping what doesn't change. The jackknife setting is basically just randomly sampling networks. This will give us the distribution we saw in the figures a couple slides ago. The mean, median, and consensus is basically different ways of computing an average graph. The consensus is a structural consensus. It was created by Rick Betzel a few years ago and has been used a lot by folks in Bratislav Misic's lab over at the MNI. I used an implementation made by his student, Ross Markello. And basically what it does is it gives you a mean graph where the ratio of short and long range connections is preserved to that what's that's observed in the words are escaping me in all the graphs that go into the average. It is the same ratio of long range and short range connections. Sorry about that. The mega case is basically we grab all the samples and we pull them together and use this super classifier with a bunch of graphs. And then the meta case is we take all the independent jackknife classifiers, as I showed again in a previous slide, something like that, and we create another classifier which learns the relationship between them. So when we have red stars, that's bad. When we have black stars, that's good. What we can see is that truncating the variance, so pretending the variance doesn't contain anything meaningful, is wrong. It makes things worse and doesn't give us anything. However, in all of these other cases, what we can find is that by actually capturing some variability, so again, jackknife is a wide distribution, so this one shouldn't really be interpreted on its own. It was just necessary for the meta-analysis. But in all these other cases, when we're capturing the variance, we actually notice we do better in prediction on an out-of-sample test set. And in some cases, we also improve the generalizability of our models, which means we essentially have closer correspondence in the performance on our validation and our testing sets. Now, this is potentially really encouraging because we tested, as I mentioned, a bunch of different configurations, and these results were pretty robust. So what we found, let's just check the time. Cool. So what we found was, again, across all of these different classifier types, SVMs, LRCs, k-nearest neighbors, random forest, and AdaBoost, we see these results pretty consistently. Most exciting to me is that this works well in AdaBoost. So AdaBoost and random forest are both ensemble classifiers. So in other words, you're already training multiple classifiers on your data. So it's exciting to me that you can create an ensemble of ensembles, and it's just kind of a hat on a hat and a hat and a hat and a hat situation. So it's interesting to see where that could potentially be pushed there. But in general, again, we have improvements kind of across the board. Validation performance is perhaps the least significant when you're thinking about ultimately the objective of creating models for biomarkers or what have you, is to be able to perform well out of sample and to have generalizable performance. In other words, you can trust the performance you get on your validation set. You can trust that it's going to perform with similar accuracy kind of in the wild. Now, one thing that was encouraging that we saw was even in cases where let's say, performance wasn't necessarily increasing or the generalizability wasn't increasing, one of them often did. So here we're showing generalizability on the x-axis and testing performance, out of sample performance on the y. So
sorry, I have a quick question. Can I interrupt? Do you mind?
Yeah, no problem at all. Sorry.
I'm not sure I got the difference between the kind of the test set and the generalizability. Like, how do you calculate this generalizability?
Yeah, so the generalizability, great question. Sorry for not being clear. The generalizability is essentially the difference in performance from our testing and validation sets. So in a perfect world, we would validate, would have our validation performance, we'd say our test has, or our classifier has, 0.8 accuracy. And then when we use this in the wild, in a perfect world, it would still have 0.8 accuracy, because then it's useful, we understand how much we can rely on this and so on. So that would be a generalizability score of 1, basically no difference. However, if in the wild it performs much worse, then our generalizability score would go down. So basically, it's the correspondence between the two. So again, if we're thinking of classifiers and models that are going to be actually operationalized in some way, actually used in some practical context, the generalizability score, as formulated kind of how I just described, is an important measure. So in this figure, in the perfect world, would be in the top right of the plots. It would be very generalizable, and we would be very performant. So in general, we want to move that way. So as I was saying, in some cases where one maybe doesn't increase, one of these features, the other one often will. So we're often moving in that direction in general. So again, here, the cluster of classifiers shown where my cursor is in purple correspond to those shown directly below them in brown for the unperturbed data. So again, in this case, the generalizability of these models didn't change. They're approximately just as generalizable as they were before. However, now they are much more performant. So again, in general, we're always getting something by augmenting our data set this way. Now, an important feature of this, one thing I mentioned ahead of time, or earlier rather, is that this is a computationally expensive technique. We're processing a lot of data because not only did I say in the dense case, we have an overhead of maybe 7 to 10 times for every execution, but we have to run our executions multiple times to get this distribution. So again, you have a massive ballooning of your computational cost. So an important relationship to understand is that between the performance benefit and the actual number of Monte Carlo perturbations we did. Encouragingly, for every method aside from our mega analytic classifiers, we found that the number of samples wasn't important. We found that there wasn't a considerable relationship between the number of perturbations and our performance. This is showing generalizability here, but for F1, there wasn't a relationship with number of samples for any of them, including mega. So again, this is potentially encouraging because what this says is maybe in reality our computational costs will double or triple or quadruple rather than being multiplied by 1,000. So the fuzzy environments that I've created is really the system that allows us to do these types of perturbations. So this exists, it's on GitHub, it's publicly available. You can Google fuzzy and my name and it'll probably be the first hit. I hope it would be the first hit. And what it allows us to really do is study essentially the stability of experiments. Again, whether in neuroimaging, as I've shown here, or other places. And this allows us to compare sources of variability that we otherwise wouldn't be able to compare. So again, we can compare things like acquisition variability, numerical error, session variability, and so on. So we can get a sense of the error bars on our results. Now, again, this is potentially encouraging because the variability we're capturing can now be used to improve our models. So instead of having single, fixed biased results from our tools, what we can have instead are, again, this distribution of highly variable results that we can try and capture. So there's many ways this can be used slightly differently or carried out in the future. So for instance, if you are more interested in tool development, you can independently perturb different steps of pipelines. Once you've done this, you can essentially consider them and evaluate them and either try and fix them in the sense of reducing the variability or at least characterize the variability of each step. You could compare the stability or uncertainty associated with different tools when deciding which one to pick. And you could also, again, perturb not necessarily pre-processing, but things like feature selection or model fitting to see the effect that may have on your essentially predictors or models as well. So again, another point that I think is important is that this isn't necessarily fixed to one part of the pipeline. There's no reason why it needs to live in this section here where we're talking about pre-processing and modeling. But this can really happen anywhere. So for instance, there's open pulse sequences and reconstruction techniques through things like QMR PulseSeg and Gadgetron. And data that's coming off the scanner could very reasonably be passed through, again, say fuzzy versions of reconstruction algorithms and Fourier transforms, so we can understand which implementations give us the most robust results. Similarly, you can test any number of tools, models, and biomarker prediction, and so on. And again, all of this can just be done by anyone at this point by launching one of the fuzzy Docker containers that's available. So I'd just like to thank my collaborators and co-authors on all this work and team. And here are some references and links.
Thanks, Greg. That was great. Does anybody have any questions or comments?
So I'm going to just put one in the chat.
Have you considered interval arithmetic, which can incorporate actual uncertainties in the raw data as well as arithmetic operation errors?
Yeah, so so in interval arithmetic, I mean, ultimately, there's still going to be limitations. Whenever you get to things like irrational numbers and fractions such as like a third or what have you, repeating decimals, it's not going to cover every base. So part of the problem with an approach like switching to interval arithmetic is that it's very involved to modify your tools and adjust them and adapt them. And it still doesn't cover every use case. You can't use interval arithmetic to represent pi. and operations thereof. So ultimately, it gets you further the same way you could, again, represent decimals as fractions instead. Many programming languages have kind of fraction types to try and avoid this. But again, ultimately, what these end up kind of doing is kicking the can. And you'll come into these errors one way or another. Thanks for the question. Looks like two Gabriels have their hands up.
I'll go first.
I think, yeah, you were first.
Really awesome, Greg. That's really cool stuff. Have you tried this at all on something a little more familiar to some of the imagers here, like an FSL tool or a registration tool or such? Yeah, so I'll show you an FSL result that I store in the back because I always get this question. So this was the proof-usurper pipeline done by HCP. It pretty much only uses FSL for alignments. And this was a study, it's actually currently under review for Mackay. And basically what we found is there's considerable variability both across operating systems and perturbed with when we're adjusting numerical error. And again, what we can expect is in a perfect world, we would have 24 or 53 significant bits of information, because that's as much as you can have in float 32 and float 64 containers. In reality, our data has something closer to 8. So there is considerable uncertainty in like the FSL library. One project I'm working on actually right now, and I paused to give this talk, is doing the same thing with AFNI. We've dug in a fair bit to FSL, and it has the exact same problems. Thanks for the question.
Okay, we can take a question from the other Gabriel.
Hey, hello. Thanks for the talk. So I was wondering if I understood correctly, all of your main results are tested on a diffusion connectivity graph.
Yeah.
So you've concentrated on the instability of, I guess, connectomic type of analysis.
Yep.
But if I think of fMRI, if we compare, I know that this is the most unreliable type of analysis in terms of like ICC reliability, where it's around 10 to 20% per connection, whereas other analysis technique can have like dual regression can go up to 0.6.
Right.
So how much have you ever considered other types of analysis, which takes a fundamentally different approach?
Yeah, that's a great question. So again, like when I showed this example here, this is structural. When I showed some of the background slides at the beginning, there's considerable variability across tools with functional. In terms of doing this exact analysis on like a functional data set, I'm trying to find functional slides here at the figure on the left. This is just the difference between tools. But yeah, when we're considering this exact kind of set of experiments on fMRI, that's something I'm working on now. And we're working with both the developers of CPAC, because, well, now they work with me at Childmind, or I work with them rather, and with the fMRI prep team. So Pierre Balek over at UAM, he's got a team and a few grad students working on essentially trying to characterize the stability of the long-term release version of fMRI prep. So we're seeing how robust it is to small changes as well. And again, the answers are pretty consistently, we see variability because again, this is just, we're perturbing machine error. And an unfortunate reality of pipelines is that in general, we aren't particularly careful with the last decimal digits we have when we're tool developers. Because in general, we tend to model something. We have, again, say a repeating fraction, 0.3 is the whole way through. And then we'll use that as though we have every bit of confidence, or 0.6s and 7s, and forget that the last 6 is kind of a 6, 6, 6, 6, not a 7. And then these errors just accumulate. It's very difficult to get around. So no, the results are pretty consistent across the others. They're just for fMRI, they're preliminary, so I didn't include them.
All right, thanks. Looking forward to see the follow-ups.
Thanks.
Dave has a follow-up, I think.
Hi again. So I was wondering whether you looked into the libraries that implement arbitrary precision in arithmetic as a way to kind of go the other direction and say, what if we cared about every single bit? Or what happens if we add more bits and more bits and more bits?
Yeah, so similar to the question kind of about integral arithmetic, what this ends up doing is kind of kicking the can. So for instance, one thing we noticed with, I'll show the FSL results that was in the appendix again. One thing we noticed was while the pipeline in general uses a lot of float 64s, in general, the results never have significance beyond what could be stored in a float 32. So even with big data types, even with storing more precision than we could, we could capture or describe with our data. Our data comes in off the scanner at int16, and we're storing it in float 64. There's never any significance up there. So kicking the can and increasing our data types, it doesn't really solve these problems. Because again, there's always uncertainty. As soon as you try and represent the decimal 0.1, for example, which is a common threshold someone may use in an experiment, or an integer multiple thereof, well, it's a repeating fraction. So whatever data type you're using in binary, it will repeat and repeat and repeat, and it'll trail off and you'll have error.
Yeah, so the arbitrary precision mathematics kind of avoids that by the way in which it's implemented for being able to properly represent these type of repeating fractions. So that's one way to do it. You bring up an interesting point about the precision coming off the scanner. And one of the challenges I've run into a lot is that even the precision you get off the scanner isn't as good as you think it is, because often the sequences are set up in such a way where you have some very, very hot voxel somewhere because of imbalance in homogeneities or receiver, the coil receiver stuff not being balanced properly, where actually you don't actually have that much dynamic range in the area of interest, where you have like a really, really hot piece of skull that wasn't properly handled during sequence control. And so you actually got much, much, much less bits of precision or dynamic range in the data itself, which is a huge challenge.
Yeah, absolutely. And I mean, even while images often come off at int16, in reality, a lot of the analog to digital converters are something more like 12 bits. So you even have less data there. So on the one hand, like using something like an arbitrary precision library, it won't solve every problem, in the sense that, again, as soon as you have an irrational number, as soon as you have an e or root 2 or a pi or whatever, which exists in all of our registration algorithms, that won't solve it. But yeah, I mean, one concept that we've talked about is essentially developing tools with declining precision models, where at each step you evaluate the precision you do have, and then you truncate your data to match that. And then that way you're not propagating uncertainty as you're continuing the process. So one thing I noticed actually that didn't end up getting included here was with FSL in particular. At the end of doing eddy correction on diffusion volumes, there ends up being a lot of kind of noise in decimal places. So you end up having, again, like kind of meaningless, irrational, like long numbers that just cascade into decimal, whereas if you just truncate that, you end up having much more stable results downstream. So declining precision models could serve as a way to try and mitigate these effects as well. But again, there's kind of no silver bullet. At the end of the day, these are just limitations of computers being inexact machines.
Yeah, I think an important thing to note on the arbitrary precision is you can go the other direction and actually limit your precision as well, which might be one way to actually implement these kinds of truncating type calculations in an efficient manner, because these libraries have optimized code for doing all the arbitrary mathematics in an optimized way.
Right.
I had a question about the expensiveness of this, way of doing things. So you said it took 20 years, correct? 20 computational years to do that?
Yeah.
So I'm wondering for the average researcher, who is, even for people outside of our institution, or people that don't have access to these compute resources, how can they get into this, or how can they start to assess the stability of their pipelines? Because as far as you saw that the sparse MCA doesn't work as well, right?
Well, so actually, that's a great question. So actually, all of the results I'm showing here, for in terms of the benefit we get with perturbations in our modeling, all of these were done on the sparse data set. So we end up seeing much more variability with sparse perturbations, but that doesn't make them worse. In fact, that may give us more opportunity to sample what is, the possible distribution of results. So in the case of, again, the sparse perturbations and the result that there's basically no relationship to the number of perturbations, you could pretty much double your computational time and get some benefit by just adding sparse perturbations that you're introducing, 2 or 3 times each. And then again, the computational overhead of the sparse perturbations to the unperturbed was something like 1.2. It was very small. Because again, in this case, I was deliberately only selecting a very small subset of operations.
Right. And would that, multiply with the number of participants that you have and everything as well? So if we're using these large data sets of 1,000 or even some have 10,000 with the UK Biobank, then would this be feasible at all?
Yeah, I mean, I kind of, the way I kind of see it is if you have the resources to process 10,000, you probably have the resources to process 20,000. Right. I mean, another thing that I didn't actually test, but would be interesting is because, these biases are definitely going to be structured in some way with respect to different tools. So even if we just ran every sample a single time, but it's a single reference time, we'd end up with somewhere in these jackknife distributions that I have here. We'd end up with something in the distributions. So it's possible that what you could do, again, is, maybe just run everything even once still with some perturbation and kind of play roulette. But I mean, in general, I think if you are interested in doing big data analysis, you probably have access to big data resources. For anyone who doesn't or doesn't know of them, Compute Canada is an excellent place to get resources. If you're a PI, you should apply. If you are a student, you should help your PI apply. Also in the States, for anyone who has an affiliation in the USA, there's this computing network called XSEDE, XSEDE. And basically they're supported by the NSF, and you can also write simple grants to them to get access to a bunch of supercomputers with, millions of CPUs. A single one of those systems has more CPUs in it than like any machine in Canada, all the machines in Canada combined. Like, they're crazy. So there's plenty of access to resources. It's just a matter of knowing where to look. So if you had that question, hopefully you wrote those down. I can write them out later.
Thanks. Are there any last questions, comments? Okay, great. Well, thanks again, Greg, for that great presentation. And if anybody wants to stick around, we'll be sticking around for a little bit of a chat with the students. So thanks again.